We're going to begin our discussions on chapter 16, which will deal with the citric acid cycle, the common mode of oxidative degradation in both eukaryotes as well as prokaryotes. Now, this cycle is also called as the TCA cycle or the Krebs cycle, TCA standing for tricarboxylic acid cycle. And this particular pathway accounts for the majority of carbohydrate, fatty acid, as well as amino acid oxidation. And as you will see in future lectures, it generates a lot of biosynthetic precursors. Now, the citric acid cycle is called an amphibolic cycle, meaning it operates as both a catabolic as well as an anabolic pathway. In chapter 14, we looked at glycolysis in detail. So you saw that glucose gets converted to pyruvate. So even though glycolysis produces two ATP molecules, it only harvests a certain amount of energy that can be found in the chemical bonds of glucose. For example, you saw that the net free energy change in glucose is about a negative 146 kilojoules per mole. However, when you completely oxidize a glucose molecule, you can generate up to a negative 2,840 kilojoules per mole of free energy. The majority of energy that is produced in the breakdown of glucose is generated by aerobic respiration. So if you remember, both glycolysis as well as gluconeogenesis was anaerobic in nature. However, as we move on to TCA or the Krebs cycle, we'll see that it involves the consumption of oxygen to generate carbon dioxide. So to provide context to this lecture, I'm going to quickly review what we did in chapter 14. So glycolysis is the first stage of oxidation of glucose. What happens is, or the end product is the generation of two pyruvate molecules. Now, under anaerobic conditions, the pyruvate is broken down to stable fermentation products. However, under aerobic conditions, the cell can use oxygen and completely oxidize glucose to carbon dioxide. And that's what we are going to discuss in this lecture. Now, this aerobic phase of glucose catabolism is also called as cellular respiration. Now, another important thing about TCA cycle is that it can also capture the energy stored in lipids and amino acids. It is assumed that this particular pathway developed about 2.5 billion years ago. This pathway is utilized by animals, plants, as well as several microorganisms, and it occurs in three major stages. So we are going to look at these individual stages in the next slide. So in stage 1, pyruvate is converted to acetyl coenzyme A. Now this is a very complex reaction that is carried out by a complex of enzymes and coenzymes called the pyruvate dehydrogenase complex. Now in aerobic organism, it is not just glucose that gets oxidized to carbon dioxide and water. So both fatty acids and amino acids also can be oxidized to carbon dioxide water through the tricarboxylic acid cycle or the citric acid cycle. Now, to accomplish this, they must first be converted to acetyl coenzyme A, because it is acetyl coenzyme A that will eventually enter the TCA or the citric acid cycle. Oh, did I mention why citric acid cycle is called as tricarboxylic acid or the TCA cycle? It is because several of the early intermediates in this pathway have 3 carboxyl groups, so that's why citric acid cycle is also called as the TCA cycle. In stage 2 of cellular respiration, the acetyl coenzyme A will actually enter the citric acid cycle. So at several steps in the citric acid cycle, a substrate gets oxidized, and in 3 of these steps, a pair of electrons is transferred from the substrate to NAD+, producing NADH, and in the fourth step, a pair of electrons is transferred from a substrate to FAD, producing FADH2. So What happens is these electrons are eventually passed from NADH and FADH through an electron transport system that is located in the inner mitochondrial membrane, and finally to the terminal acceptor, which is oxygen. So that would be stage 3, which is electron transfer and oxidative phosphorylation. And it is this step that generates the vast majority of the ATP during the catabolism of glucose, fatty acids, and amino acids. Let's take a closer look at the stage 1 of cellular respiration, which is the conversion of pyruvate to acetyl coenzyme A. So the net reaction is the oxidative decarboxylation of pyruvate. This is an irreversible oxidation product process where the carboxyl group of pyruvate is actually removed as a molecule of carbon dioxide, and the two remaining carbon atoms, they eventually become the acetyl group of acetyl coenzyme A, which is this entire molecule that is represented here. Now, this is a very favorable reaction. That is why you see just one arrow going this way. It is an irreversible process. Now, we also remove a pair of electron or 2 electrons from the pyruvate molecule. So you would never have free-floating electrons. Instead, they're found as what you call a hydride ion. So you have the 2 electrons and a hydride ion. Now, this hydride ion will be accepted by our electron carrier, NAD+, resulting in the formation of NADH. So you have a molecule of carbon dioxide and 2 pairs of electrons, or a hydride ion that is formed, which will result in the reduction of NAD+ to NADH. So we saw that pyruvate get converted to a 2-carbon acetyl group. However, this acetyl group must be activated to enter the reactions of the citric acid cycle. The activation happens when the acetyl group is bonded to the thiol group of the coenzyme A. So this is your acetyl group, and it has to be bonded to the thiol group of a coenzyme A. Now, coenzyme A is a large thiol that is synthesized from ATP, pantothenic acid, or vitamin B5. So you have your ATP here, the pantothenic acid, and a beta-mercaptoethylamide. Now, the coenzyme A and the acetyl group gets bonded at this highly reactive thiol group to form a very high energy thioester bond. So technically, let me get my eraser so I can show you what we're talking about. So this is a thioester bond, and I'm going to put a squiggle here to show that it is a really high energy bond. So and the resulting molecule is called the acetyl coenzyme A. So it is acetyl coenzyme A, also called as the activated form of acetyl group, that enter the citric acid cycle. Now, as I mentioned earlier, the conversion of pyruvate to acetyl coenzyme A is catalyzed by a multi-enzyme complex called pyruvate dehydrogenase complex, or PDC. So as a general rule, multi-enzyme complexes are groups of enzymes that are non-covalently associated to each other and catalyze at least 2 or more sequential steps in a metabolic pathway. So in this case, you saw there over here that it has 3 different enzymes, which is E1, E2, and E3. So let's take a look at what these enzymes are. So E1 is a pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. Now, there are several advantages to having multi-enzyme complexes catalyze a reaction. For example, you have learned in, I think, chapter 6 of Biochem 1 that enzymatic reaction rates are limited by the frequency at which enzymes collide with their substrates. So if successive metabolic reactions happen within a multi-enzyme complex, the distance between the active sites are reduced considerably, enabling the substrate to diffuse efficiently and thereby enhancing the rate of a reaction. This process of reducing the distance that the substrate must diffuse to travel from one active site to another is called channeling. Now, channeling also ensures that the side chain reactions are minimized and provide a very efficient way to coordinately control this reaction. So pyruvate dehydrogenase complex can catalyze 5 sequential reactions in the conversion of pyruvate to acetyl coenzyme A. Now, there are several coenzymes and prosthetic groups that are required for this sequential reaction to happen. So the first one is thymine pyrophosphate. Let me highlight this really quickly in our slide. So, it is already highlighted. I've just circled it. Now, the second one is nicotinamide adenine 9-nucleotide or NAD+. The third one is FAD or flavin adenine dinucleotide. It's right here. And the fourth one is called lipoamide. So it is represented as a lipoyl lysine between its two different forms, which is the reduced and the oxidized. And so let us take a look at lipoamide in detail. So I'm going to go back and pull this slide. So a lipoamide is a lipoic acid that is joined by an amide linkage to the epsilon amino group of a lysine residue of E2 enzyme. So let's take a look at this. So together, this lipoic acid that is hooked by an amide linkage to a lysine residue of E2 is called a lipoamide or a lipoamide. Lipoamide. I don't know how to pronounce it. All right. So now this lipoamide can exist in 3 different forms: the oxidized form, its reduced form, and an acetylated form. So in its oxidized form, it has a cyclic disulfide group. In its reduced form, it acts as a carrier of hydrogen atom, and in its acetylated form, it can act as a carrier of acetyl group. So this is important to shuttle electrons and acetyl groups in the sequential reactions you will see in a minute. The reaction starts with the pyruvate, and although we say that it is the conversion of pyruvate to acetyl coenzyme A, there are 2 products that are actually made. So it's the conversion of pyruvate to acetyl coenzyme A and NADH. So in step 1, you decarboxylate pyruvate to form an aldehyde. In the second step, you oxidize this aldehyde to form a carboxylic acid. In the third step, you actually have acetyl coenzyme A formation. In the fourth step, you reoxidize the lipamide cofactor. And in step 5, you regenerate the FAD cofactor back to its oxidized state. So there are 5 overall steps. So due to time constraints for this video, I'm going to stop here. However, then in the next video, we will go over these reactions in detail.